I already hit record, but you can grab. Where are the tissues? They're gone here. They're gone there. Yeah, I don't know. Okay. All right. Somebody remind me of the 5 phases of mitosis that we left off on Friday.
Non-dividing cell.
So that's a non-mitotic. Oh, you're looking at this? So, prophase. We're gonna start there.
You're so smart.
Okay, so prophase. By the time that you begin prophase, you're in it. You are in mitosis. And mitosis is one of these things that once it starts, it's going to finish. Unless it fails at a particular checkpoint, something goes wrong. Once it starts, it finishes. Okay? So once it starts, mitosis, it's going to finish. Unless there's some mistake and the cell needs to just shut things down.
Which is like recombine.
Oh, we'll show you. We'll show you. Yeah, Mark.
Wait, I know the next slide.
Okay, well let's work on prophase for a minute and then we'll go to the next phase. Okay.
These are phases of the cell cycle.
These are phases of just mitosis, which is just one portion of the cell cycle, right? You've got that pie drawn. So in prophase, there are 2 big things that you can see going on if you look at the cell. One is the chromosomes condense. So when you think of a chromosome, what do you see? An X, right? And we saw this last time, right? Something like this. Okay? Chromosomes do not look like that when the cell is just carrying out its normal cellular functions. You can't actually see the chromosomes because they aren't condensed enough for you to see them using light microscopy. They're barely condensed enough to be able to see them using like electron microscopy, but yeah.
Okay, so what is it like Down syndrome? Do you have an extra pair?
You have either an entire extra copy of chromosome 21, or you can just have a portion of extra.
And is that in like
21.
all your cells?
Yes.
Oh, wow.
Yep. And so it creates a gene expression problem, but we'll talk a lot about gene expression in the coming weeks. So during prophase, the chromosomes become visible. Okay? So that's something you want to look for. And another one is what we call the mitotic spindle. Bless you. The mitotic spindle forms. And you're like, Dr. Engel, that mitotic spindle sounds wonderful. What is it? The table.
Something at the end. That, like,
Yeah, it's yes. So also during this phase, but this is something that's difficult to see, is the centrosomes migrate to opposite poles. So these are the big events, if you will, that happen during prophase. These 2 you can see when you're looking at a cell. This one, you can't really see that migration, but it does happen during prophase. So the mitotic spindle, thank you. Centrosome. Centromere is on the chromosome. This right here, where these 2 chromatids are attached, is the centromere, but the centrosome. And so the mitotic spindle and the centrosomes are connected, because the mitotic spindles grow out from the centrosome. So I'm gonna draw some arrows over here, and I'm gonna illustrate to you what's going on. All right?
Awesome.
Yeah.
I can't wait for this.
So here's the cell, and the centrosomes are gonna migrate to opposite poles. So here's one centrosome, and here's the other centrosome. And you're like, wow, those are weird looking structures, and they kind of are. They're basically 2 cylinders that are perpendicular to one another, and they're physically attached together. But out from here grow the microtubules that are needed to separate the chromosomes. And so here I'm going to change colors here. I'm going to make this nice.
Ooh.
Can anybody struggle to see green?
No, just do it. Baking stuff.
Green is a big blue.
Those are the spindle fibers.
Yep, spindle fibers. So we have formed the mitotic spindle. This is the mitotic spindle. And the mitotic spindles grow out from the centrosomes that migrate to opposite poles during prophase. And you're like, that's so cool. And you know from when you learned this previously that these spindle fibers are what are going to actually separate the chromosomes during this process. Right? They're also going to help elongate the cell to ensure that as they separate, they end up far enough apart that you form two separate nuclei. Okay? But here's our mitotic spindle. Here's one spindle, here's another spindle, and they grow out from those centrosomes. Okay? This is all during prophase. Okay. Now, this isn't necessarily what prophase looks like because I didn't draw any chromosomes, right? So you look at this and you're like, Dr. Eagle, where are the chromosomes? You said that chromosomes become visible during prophase. I just was drawing the mitotic spindle. Relax. Okay? Relax. All right, any questions about prophase?
Oh, what are the little butts?
The centrosomes.
Oh, okay.
Yeah. Centrosomes. And then they're made up of two centrioles. Make up the centrosome. Okay? Any other questions about prophase? Or the mitotic spindle? Yeah. Sure.
What was the second part of the mitotic?
The mitotic spindle? This is the mitotic spindle.
Oh, what is it for?
What is it for? It's to separate the chromosomes and to lengthen the cell. Okay? Alright. Okay? Any other questions? Because I'm going to erase this and prophase. And Mark's going to give us our next stage.
I didn't think the next stage was the other stage.
Mark, what's the next phase?
Prometaphase.
Prometaphase. Prometaphase. Now, when you're actually looking at cells, it is very, very difficult to tell the difference between prophase and prometaphase. So when I give you a little task to work on in a little bit, when you're working with cells and you're counting, we're going to lump these two phases together, okay? Because it's hard to tell them apart looking at cells. It's not hard to remember what's different, okay? So there are two big things that happen during prometaphase. You ready? Two big things. The nuclear envelope dissolves. Let's instead of dissolves, let's say breaks apart. Okay, so the nuclear envelope, what's forming the compartment or the wall around where all those chromosomes are, it breaks apart during prometaphase.
Where does it go?
Where does it go? That's an excellent question. It basically goes into small little vesicles, right? So we've talked about that before, membranes breaking down into vesicles, right? So you can move materials. But the nuclear envelope breaks down into vesicles and just stays that way until it reassembles at the end. Yeah. Okay, so that's the big one. And then some microtubules, and this comes from the mitotic spindle. That's what those spindle fibers are. They are microtubules. Okay, microtubules from the mitotic spindle. Those mitotic fibers, that makes sense? Okay, contact the chromosomes. Okay, the microtubules from the mitotic spindle contact the chromosomes. And I want to make a specific point here. One from each pole. Okay? So the microtubules contact chromosomes, one microtubule from each pole. Okay? So I'm going to draw the mitotic spindle again, but I'm not going to draw the full spindle. I just want to show you a little bit what this looks like. So we need a different color. Orange, that's not going to work. Okay, I'll just use green again. So here's our cell. Here's our chromosome, right? Here's our centrosome. Here's our other centrosome. And here is a microtubule from each pole that makes contact with that chromosome. And Kyle, what location on the chromosome do they make contact with? The centromere. Ah. That region of the chromosome. Now, what you find out when you look at this in a lot more detail that it's not quite this simple, but we can leave it this simple for now. Okay? If you all decide to go and to study biology in college, you'll realize it's a little bit more complicated than that. It's okay, Isa.
The spindles are those lines, right?
Yes.
Okay.
The spindle fibers. The spindle is the whole thing. The spindle is the centrosome with all of the fibers. But this is a spindle fiber, which is a microtubule. Okay? So this happens during prometaphase. This can't happen unless the nuclear envelope dissolves. Although, actually, in like dinoflagellates, but we'll get there in a minute. In dinoflagellates, the nucleus never dissolves. They actually grow through, the microtubules grow through the nucleus. And then in diatoms, which are another type of organism, the mitotic spindles form inside the nucleus. Anyways,
Wait, do the plant cells...
Plant cells do this as well. Plant cells, animal cells, fungal cells, almost all eukaryotes do it this way, but then there are some dinoflagellates, where the nucleus never dissolves, the spindle fibers grow through the nuclear envelope, and then diatoms where the spindle forms inside the nucleus. And other than that, they all basically operate like this: plants, animals, fungi. Yeah, Emma. Some yeast, which are single-celled fungal organisms, do things like dinoflagellates, where it'll grow through the nuclear envelope. The nucleus never dissolves. Okay. All right. So that's prometaphase. Yeah, Katie.
Can you get more tissues?
Yeah. Where are you gonna go?
To the front desk.
To the front desk.
Yeah,
The front
with tissues.
Tell them I said hi.
Okay.
Thanks. Okay. So that's prometaphase. Any questions there? Okay. So again, when you're looking at a cell under a microscope, it's very difficult to tell this apart from prophase. Okay, so we'll lump them together in one when you actually start looking at these under the microscope. All right? No questions? Thoughts? Concerns? Okay, again, we're saying like this is the norm. There are some exceptions to this. Again, those dinoflagellates where the nuclear envelope doesn't dissolve, some yeast organisms, and then diatoms where the spindle forms inside the nucleus. Okay? Wow. For a Tuesday, you guys are unusually tired looking. Mondays, it happens. And I don't mean you look tired like you look like it's like a nice way of saying you look sick. I mean like you really just look like you want to go to sleep.
Yeah.
Yeah. All right.
It's a fair assessment.
But not like you just woke up tired, but like the end of the day tired. You know, does that make sense?
Yeah.
To distinguish? Okay.
I just look sick. I have taken the acupuncture.
Because like you just woke up tired, you look sloppy. You don't look sloppy. It's like the end of the day. The end of a good day. Is that helping? Did I offend you? Okay.
No. It's like right in the middle.
Alright, metaphase.
Yeah.
Now in metaphase, I'm gonna give you one feature of metaphase that you need to know. Okay, just one. That's it. Chromosomes line up. Whoa, you line up in the middle of the cell. Sometimes we call this middle the equator. Sometimes you'll see it referred to as the mitotic equator. Sometimes you'll see it referred to as the mitotic midpoint or the cellular midpoint. But ultimately, the chromosomes, sorry, that needs to be, you can't have a line with one person, right? Chromosomes, you're like, these aren't people at all. You can't have a line with one chromosome. Although that would be wonderful to go to Disneyland and have a line of one person, right?
She's going to Disneyland on Monday.
Today is Disney Day at Masters, and so there are like 300 Masters students descending on Disneyland. Why didn't I get?
This is why.
Because I'm here.
Because this
Plus, if I'm gonna go to Disneyland, I'm not gonna go with a bunch of college students. I'm gonna take my family, and my kids are in school. And all right,
I've been to Disneyland every
you know, do I make fun of my kids often?
No.
Oh, it must be just at the college where I make fun of my kids. Because I was gonna say I really do enjoy spending time with my kids, but you all wouldn't think otherwise. But I make fun of my kids a lot.
I mean, sometimes.
You've made fun of them like twice.
Yeah. So chromosomes line up in the middle of the cell. That's the hallmark feature of metaphase. And you can see this. This is very obvious when you look at a cell. If it's in metaphase, all the chromosomes are lined up, single file. Now, some of the chromosomes are longer than others and look sloppy, but you can still tell that they're in line. Right? It's just like, you know, there are things draping off them. It's like when kindergartners line up. Have you ever watched the kindergartners line up?
They're actually better lining up the numbers.
I believe that to be true, but they're just kind of sloppy. Like some of them are dragging their backpacks because it's too big to have on their shoulders. Right? Their coats, their mom didn't zip their coat up, so like their clothes are falling off and they can't figure out how to do it themselves. Man, it's just so it's sloppy, but it's alive. Easy.
Where in the cell does mitosis happen?
Where in the cell?
In the cell.
The entire cell, all of the space of the cell is devoted towards carrying out mitosis.
No.
Yeah, it only happens during a small portion of the cell cycle. But yeah, the entire cell is committed to carrying out
So what
mitosis.
about all the other parts in the cell? They're just kind of sitting there while everything's happening.
You're talking about like the other organelles and stuff? Yeah, I mean they're basically being displaced, pushed out of the way to ensure that the
And what about all the ribosomes? Do
DNA.
they float around?
Yeah.
Interesting. I didn't take bio.
Lydia. Well, nobody usually asks those kinds of questions. Those are really good questions to ask. Like Rick last time, he's like, well, don't all the organelles need to be duplicated as well? And they do, but again, nobody really asks these kinds of questions because they're like, okay, this is enough for me, right, of what's going on, and they don't think about like what's everything else in the cell doing. I mean, it's still there. It has to be kind of displaced and pushed out of the way. It's pushed out of the way by the mitotic spindle. The mitotic spindle is kind of pushing everything out of the way to ensure that the chromosomes are separate. Yeah, that mitosis can happen. Okay, shh. Because remember, prior to mitosis, you had S phase, right? And what happens during S phase?
What do you mean?
The DNA got duplicated. So once the DNA gets duplicated, you have to go through mitosis. Otherwise, you have twice the amount of DNA you're supposed to. And you all know, you brought up Down syndrome, right? If you have just a small extra portion of chromosome 21, you could have just like one extra gene. So you have one gene that instead of 2 copies, you have 3. And it leads to some completely different developmental processes, right? So imagine if you had 3 copies of the entire genome instead of 2. It'd be catastrophic, right? Let alone 4 copies that you have after S phase, right? You have 4 copies of the genome. So now you have to go through mitosis to separate those 4 copies into 2 separate cells. Emma and then Isa.
What if you're like an extra copy of chromosome 92?
So there are very few chromosomes where you can have an extra copy and even survive the developmental process. There are very few of them. So chromosome 21 is one. I think chromosome 19 as well, and there's another genetic illness associated with it. You probably can't have an entire chromosome 19. And then there's trisomy 13, where you have an extra chromosome 13. But it's not, having an extra chromosome is usually catastrophic for development. Because it's too hard for the cell to control how much product they're making.
What if you're missing one?
If you're missing one, it depends on which one you're missing. Sometimes that can be that burden can be shouldered more easily than having an extra copy.
Wait, what do the chromosomes do? Like,
No, they contain genes that code for proteins, and they also contain gene that code for regulatory elements that control expression.
my question was, how does chemo,
Chemotherapy work to it targets and kills cancer cells.
does it?
But there's some what's the term you use? There's some yeah, there are obviously side effects, and a lot of those have to do with gosh, there's a term that we use in warfare. Not friendly fire, but when you accidentally kill civilians in the process of trying to kill the
I know
enemy.
what you're talking about.
I don't know what we're talking about.
What's I had
Anyways, there's when in the midst of trying to kill
damage?
yes, collateral damage. In the midst of trying to kill the cancer cells, you kill some normal cells as well,
like your motors, like
Yeah.
your motor.
which is where a lot of the side effects come from. All right. Any questions on metaphase? So this is very obvious when you look at this under a microscope. If they're lined up, it's metaphase. Okay.
Wait, lined up horizontally or vertically?
Well, it depends on the way the cell's oriented.
Wait, so we're gonna draw it?
You'll see it.
Oh.
Yeah, you'll see it. They're very obviously lined up, but it depends on how the cell is oriented. Remember, cells exist in 3 dimensions, so it's not quite so easy as all the cells are. Yeah.
Oh, so,
Yeah. It's not so simple as like they're always oriented the same exact way.
I don't know.
All right, next one. Next phase. Next phase. Anaphase.
Metaphase only had one underneath it?
Yep, just one. Okay, anaphase. This I'm going to give you two.
How come we didn't do
Two events.
Prometaphase after? Or wait, we already did.
Yeah.
Okay.
So what we call kinetochore, and I'll put in parentheses what this means. Kinetochore, these are the microtubules that are attached to chromosomes.
Wait, higher
Kinetochore. Need to close my parentheses. So, can you even read chromosomes over there on the? Okay. So kinetochore attached to the chromosomes, microtubules shorten. Okay. The kinetochore, those that are attached to the chromosomes, microtubules shorten, and this separates. Well, I don't know why I did a cursive S. Separates the chromatids. And I will draw this one for you. At the same time, polar microtubules,
There's only one thing.
polar microtubules, which are still, these are still part of the mitotic spindle. Okay, the kinetochore microtubules, those are attached to chromosomes, shorten. This separates the chromatids. Polar microtubules lengthen the cell. And I'm going to draw this for you so you can see both these types of microtubules, and they are both growing out from the centrosomes. They are both part of the mitotic spindle. Okay, so here is a kinetochore microtubule. It's attached to the chromosome. These shorten, which pulls these two chromatids apart. Okay, those chromatids were both part of this chromosome. They happen after the DNA is duplicated. And then at the same time, we've got other microtubules. Called polar microtubules. You see they're still growing out of these centrosomes? You see? Still part, bless you, still part of the mitotic spindle. They are lengthening the cell at the same time that these are shortening and pulling those chromatids apart.
Wait, what are the little
These motor proteins functioning to actually walk those in separate directions. Man, I love that you all are like, wait, what's really going on here? Most people would just be like, okay, this is fine, this is enough for me.
Wait, so can you explain them
Never.
one more time? Like explain what everything's
Never
doing?
stops.
Yeah, so here, these are kinetochore microtubules. They're physically attached to the chromosome. They are shortening, which pulls, so these, I don't know if I mentioned this on Friday, but these are called sister chromatids. Oh yeah, I did. Because I remember I wrote this and then somebody was like, sister taxa.
Oh yeah,
Right?
that's me.
Yeah. So the sister chromatids, they should be identical, because they're the two copies of the genome. So like, let's say this is your chromosome 1 from mom. During S phase, chromosome 1 got duplicated, and both copies are attached together at the centromere. Okay. Well, they are detached from each other during anaphase as these kinetochore microtubules shorten. And then at the same time, these polar microtubules lengthen the cell. This is another one that's very obvious when you look at these under a microscope, that the cell is in anaphase. Because the cell is, instead of being roughly circular, it's very elongated. It looks like a football. Issa and Katie.
So the sister thing. So if two twin sisters married two twin brothers, their kids would have a way bigger.
They would have their kids would be unusually highly related.
Yeah.
But that would happen. So their kids would be more related than typical cousins are. They'd be more similar genetically.
Because their dads are brothers and
But that would be true even if, so typically siblings are half of their genetic material is the same, right? We talked about this early in the year, okay? Right. Cousins are typically 1/8. 1/8 of your what's the term I want to use? 1/8 of your particular copies of the gene, your particular versions of the gene are going to be matched with a cousin. Okay? But if your cousin, even if your cousin was just from an identical twin, like your mom and your aunt were identical twins, instead of being 1/8, you would be 1/4, which would be typical for half siblings, right? That share one parent, but I mean, so even that. So then, if you actually had your moms were identical twins and your dads were identical twins, you would be 1/2. It'd be like being a full sibling, even though you're cousins.
Whoa.
Yeah.
That's crazy. I wonder if that exists anywhere.
Oh yeah, it happens a lot. Identical sets of identical twins get married fairly regularly, and
We gotta do it. Usually the two
Alright.
would be actually the two are
imagine identical triplets, right? And you get 3 triplets marry 3 triplets. Okay. All right.
That's wild.
Any other questions on anaphase?
Okay, what are the black things?
These?
Yeah.
Those are motor proteins that are forcing basically these motor proteins will force these microtubules to move in opposite directions. And will help to lengthen the cell.
What happens when they lengthen too far? Can they lengthen outside?
Well, the whole spindle will be broken down before that. Because the very next phase, one of the things is mitotic spindle disappears, nuclear envelope reforms, chromosomes decommits. Yeah.
What? Wait.
Oh, sorry, it was Issa and then you, and then I forgot. I'm sorry.
It's okay, I forgot to.
Okay.
I'll ask again.
All right. Any questions on anaphase? Yeah.
I remember. Okay, so when you look at it under a microscope, how can you see inside the cell?
Because they've stained the chromosomes. So you
So
can.
it makes it clear or something?
Well, I mean, the cells, light can pass through cell membranes very easily, so you can usually see into a cell when you're looking at it under a microscope, because light's going to pass through. And then they usually would just look completely colorless. Everything inside would look colorless, but if you stain the chromosomes, you could see them because they'll absorb some of that light, but not all of it, right? So if you stain them blue, they'll absorb all of the light except for the blue portion of the visible spectrum.
Cool.
Yeah. All right. So this is disappearing. We've only got one more phase. What is it?
Oh, I know it.
Yeah. What is it, Issa? It was you, Issa, right? That were saying you knew it.
She did say it. She just
Oh, you did.
got a deeper voice.
Yeah. Okay. All right. Now I will tell you, telophase is hard to distinguish from just having two cells at prophase. Telophase?
Oh, so it's not telophase, it's telo?
Telo, I don't know. I've heard both.
Telekinesis.
A lot of people say telophase. I like to make it a hard E though.
Oh my god.
So the mitotic spindle dissolves. The mitotic spindle dissolves. The nuclear envelope reforms. But instead of reforming as a single nuclear envelope, the nuclear envelope reforms as two nuclei. One around each pole, right? The chromatids got separated to opposite poles. And so now when the nuclear envelope reforms, it doesn't reform as a single one around all the chromosomes. It reforms around as two nuclei, one at each pole. Okay?
Yep.
Alright. And then the last thing, chromosomes decondense. Chromosomes decondense. Okay, so those are the big things that happen during telophase or telophase.
This is easier than like cell communication.
Yes.
By far.
Yep. Well, one, you've seen it multiple times before. You probably saw it in junior high when you did life science in junior high, and then you saw it in honors bio.
That was a class.
Isn't it chromosomes?
That was a
What was that?
class.
Or it has to be chromosomes a little bit.
In the front?
Oh, I know you're talking about.
Subject verb agreement. Thank you, Tori.
7th grade.
Chromosomes decondense. Okay? So, real quickly, I'll give you a clue to how to distinguish this telophase from prophase. So, I mean, the chromosomes are in the process of the chromosomes decondensing, they're going to look the same as in the process of condensing, right?
Wait, can
You
we
have
draw it?
what's that?
Are we gonna draw this one or no?
Sure. Bless you. Now your chromosome doesn't look like this because your two chromatids have been separated, one into each pole. Your nuclear envelope reformed, one around each of those poles, and your chromosomes begin decondensing. Look, the mitotic spindle is gone. Now, the reason why this is hard to distinguish, you're like, Dr. Engel, that should be very easy. If you see a cell with two nuclei, it's in telophase. But at the same time that anaphase and telophase are going on, there's another process happening. Look to your cell cycle, your pi, and what is M phase? It says mitosis and cytokinesis. So at the same time that anaphase and telophase are happening, the cell is actually dividing into two cells. So it's very, very difficult to find a cell with two nuclei. It's almost impossible because at the same time that this is happening, the cells are dividing into two cells.
Is there
So-
like a, oh, sorry.
Oh, go ahead.
Is there like a split second where like the cell is open?
In the middle? Yes.
So does
And the
that-
two cytoplasms are actually connected for a little bit? Yeah.
Nothing gets in though?
I mean, they were originally one cell, so they're in the process of splitting everything in half. You know? Things get split pretty evenly, unless they are purposely not split evenly. But we'll talk about that when we talk about meiosis on Thursday. Because on Thursday we need to talk about meiosis and gamete formation. Like what happens, because if I see you, Trinity, and we'll come in just a second. So mitosis works really well to make two identical cells, right? You have one cell, you need to make two because you want to grow or you want to develop or you need to repair damage. But it doesn't work so well for generating cells that can then be involved in sexual reproduction. Because if you took one of your cells, if it was produced from mitosis, it has a full copy of the DNA, right? It actually has two copies of the DNA, right? Just every cell in your body, right? And if you took that and fused it with a cell from somebody else, now there's 4 copies of the DNA. It's not good. So you need to generate cells that only have 1 copy, that have half the amount of DNA. We do that through meiosis. Trinity.
Ready to draw a cell for metaphase?
Oh, because you want me to draw one for, I'll draw one for metaphase. Well, you're the second person that's asked. Okay, so this cell I'm about to draw is for metaphase, it's not for telophase, even though telophase is on the board, okay?
Oh wait, no, this is metaphase.
Yep, that's metaphase. They're lined up in the middle of the cell.
And it's still pretty circular.
Yes. So it's almost impossible to catch a cell in the process of having 2 nuclei. If you do, you know it's telophase. Okay, but usually they've already split into 2 cells, so the way you would tell them apart is you would count this as 1 cell in telophase, and the cells are much smaller than the ones that are in prophase. Because they haven't gone through G1 where they've grown a lot. Okay? So you can look at it based on size. Because I'm going to task you with taking cells and finding 100 total cells that are in this actively mitotic tissues and counting the number that are in each of these phases. Okay? So any questions on this?
No.
Okay. This is going away. Because there's always the question of, well, how long does this take? Right? And how long does the cell spend in each section? And so we have a little trick to figure this out. So the recording is started again, and want to make sure that we get these data. So this, again, this is for whitefish blastula. I don't think anybody that used the plant cell was able to count any data. So we had 3 groups with numbers for interphase. This group right here, how many cells did you have in interphase?
Like
It's at 50 plus?
50 plus.
So let's say
Should we say
60.
chapter? Okay.
Yeah, should we just say 50?
Sure.
Okay, so we'll say,
We got theirs, and then average
50.
theirs with 50.
well, we're gonna add them all together. 50, ladies, what'd you get? 0 in interphase? Oh, so you didn't count those?
I don't know.
Because those are any ones where you can't see the purple marks. So we'll say 50 for you as well, and then over here, 51. So 151 cells in interphase. All right, prophase / prometaphase. 8, ladies? / prometaphase.
44.
44. Okay, so 44 and 8 is 52? And then what'd you have? 11. 52 and 11 is 63. Okay, metaphase. 2?
16.
16. So that's 18. 23? Thanks.
If 18 plus 7.
Oh,
20. Yeah, 25. Oh man, I love it. All right, anaphase. 7. 2, 9, 11. Telophase. 2. So that's 16. 29. So 16 and 29 is 30. 35?
Yeah.
Okay.
45.
45. Thank you. All right. So total number of cells, 151 and 63 is 214, 239, 250, 295. 295 total cells. Okay? 151 out of 295 is approximately 50%. Right? Because 150 out of 300 would be 50%. It's a little bit more than 50%, but we'll say approximately 50%. 60 in approximately 300 is going to be about 20%. And by the way, these abouts, it may end up being more than 100%, but we're approximating, so it's okay. Right? It's not ideal, but it's okay. 25 in 30, let's see, 1 in 4, 1 in 12. So the 1 in 12 is about 8%, maybe?
Yeah.
Okay, so we'll say about 8%. 11 out of 295 would be about 4%. And then 45 out of 295 would be about 15%. Okay, so that's 15. 15 and 20 is 35. 35 and 12 is 47, and then 50 is we're close to 100%. Okay, so then what this means is, according to these data, which I agree that you had a lot more cells in M phase than these two groups did. And so your blastula was just growing more rapidly. You probably had the blastula, or the slice of blastula you had was just growing like crazy. Okay? But according to these data, if this entire process to work through the entire cell cycle took 5 hours, about 2.5 hours would be spent in interphase, right? About, what's 20% of 5 hours? 1 hour? About 1 hour would be spent in prophase / pro metaphase. About, about a little bit less than half an hour would be spent in metaphase. A little bit less than 15 minutes spent in anaphase, and about 45 minutes or so spent in telophase. Okay?
That's pretty cool.
All right? So when you look at these data, what does it tell you about the length of interphase? It's long, right? And that makes sense because interphase is made up of 3 phases: G1, S, and G2. And in S phase, you have to replicate all of the DNA. In G1 and G2, you have to grow and prepare for the next cycle. Okay, the next phase in the cycle. Make sense?
Yeah.
Okay, so where you can anticipate seeing this on the AP exam is you may see these data presented. You might see like somebody did this process, they present these data to you, and then they ask you questions about the length of each phase. Okay. And they may ask you a question like, let's say you could speed up this phase, what would the overall impact on the cell cycle? Alright. Any questions here? We have one less concept from chapter 12, we need to talk about it, and then we'll be done. And on Thursday we can do chapter 13, which I believe is on meiosis, but that I'm just assuming. I don't remember. Okay? Alright. Okay. So remember on Friday, I gave you 3 pieces of data, and the first one said most cells are not currently in the cell cycle. They are not dividing. They've left the cell cycle, right? Do you remember I told you that? And then so what we need to do is then figure out how does a cell progress through the cell cycle, okay? And so we have what are called checkpoints. So draw your cell cycle again. Thank you. Did you just tell somebody they look like SpongeBob?
That looks like, leave a circle in SpongeBob where it draws that really fancy person and erases it. And Squidward can't draw it.
I don't, I'm sorry. Okay, so this is G1, this is S, this is G2, and this is M. And in this cell cycle, there are 3 checkpoints. Okay? Where's my green?
Who's mad?
Sorry to interrupt.
Yeah.
Just looking for fire extinguisher.
Oh, yeah, no worries.
Somewhere around here.
maybe under this sink.
Was there
No.
The lady said it was on here. I apologize.
No, no worries. I have no idea where it is.
I'll come back
Okay.
if there's one in there.
Okay. So there is a checkpoint here called the G1 checkpoint. G1 checkpoint. There is a checkpoint here called, what do you think it's called?
G2
The G2 checkpoint. And there is a checkpoint here. What do you think it's called?
M-phase checkpoint.
It's just called the M checkpoint.
What are these checkpoints?
That is a wonderful question. We're going to talk about that in just a second. Okay? So we've got 3 checkpoints here. At each of these checkpoints, you are looking for 2 things. At each checkpoint, okay? Doesn't matter if it's G1, if it's G2, or if it's M, you're looking for 2 things. 1, so at checkpoints. Doesn't matter which checkpoint it is, again, you have 2 things. You're checking for errors. Checking for errors. Okay, in G1 and G2, those errors are usually going to be DNA errors. There are errors in the code, okay, that trigger, proteins at this checkpoint to do something and result. Okay. Okay, so first thing is you are checking for errors. The second thing is you are checking for the machinery needed for the next phase.
Why isn't there an S for it?
That's a good question. They're just because what the G2 checkpoint will check for is to ensure that the S phase was done correctly. The main reason why there isn't an S checkpoint is because during S phase the only thing happening is duplicating the DNA. Everything else is pretty much shut down, including because all of this work has to be done by what? What does the work of a cell? Thinking about the analogy of a factory, yeah, the proteins are doing the work of a cell, so you need active proteins. So you're checking for errors, and you're also checking for the machinery needed for the next phase. If either of those fail, the cell does not progress through. Okay? If either of these checks fail, then the cell does not move past that checkpoint. So which tells you most of the cells in your body did not pass which checkpoint? If they've left the cell cycle entirely. G2? The G1. Because if they went to G2, they've duplicated the DNA and they can't function properly, right? We talked about that. Extra DNA is bad. Yes? Extra DNA is bad. Most cells in our body failed the G1 checkpoint. And so they have not progressed past it. And so they're basically stuck here. And we call that being in G0. It's not really G1 because G1 leads to S, right? But G0, it failed the checkpoint. It left the cell cycle entirely. Until what happened to trigger it to go back in? We talked about this on Friday. What does it need? What does it need to re-enter the cell cycle? Growth factors, right? And remember, in cancer cells, they can do that in any environment, so they're making their own growth factors. Okay? Emma, you had a question?
Yeah, so say for example, someone finds cancer at a really, really early stage, and they just individually remove the cells.
Yes. That's what you do when you surgically remove a tumor before it has a chance to metastasize and cells start breaking off.
Can they do like one by one?
Yeah, sure. But it's painstaking. It's easier to just remove some healthy tissue and allow mitosis to repair it.
Because like sometimes the tumor is like close to like in your brain, it's like
Sure.
dangerous to remove the entire cells. Can they just
Yes, remove a portion of it and then
just grow back?
yes. Yeah, and so some types of cancers, if the tumor's in a particularly dangerous place, you just regularly have to have some of the tumor removed. And And then they'll try to kill the rest of it with radiation and chemotherapy. Okay, so you check for errors and you check for the machinery needed in the next phase. The machinery needed in the next phase is always going to be this. It's going to be a cyclin dependent kinase.
Wait, what is this part?
This is what the machinery needed for the next phase. The machinery needed for the next phase at all of these checkpoints is some cyclin-dependent kinase plus cyclin. Right? That makes sense. If it's cyclin-dependent kinase, it only works in the presence of what? Cyclin. Okay? So every checkpoint, the particular type of cyclin-dependent kinase is different, and the particular cyclin is different, but it's always a cyclin-dependent kinase plus some type of cyclin. Cyclin is a protein that activates cyclin-dependent kinases. They call it cyclin because the concentration of it varies throughout the cell cycle. That's why they call these proteins collectively cyclin proteins, because their concentration varies throughout the cell cycle. So for instance, to get past G1, you need cyclin-dependent kinase 4 and cyclin D. Okay? And so cyclin D, it's going to get really highly concentrated here, but then it disappears in the rest of the cell cycle. Okay? And that's why, again, it's called cyclin, because its concentration varies throughout the cycle. It's cyclin, you know? Sorry. All right, what is a kinase? We've seen this type of enzyme before. We saw it throughout glycolysis. We saw it in photosynthesis. What is a kinase? Emma.
Oh.
Trinity. It's an enzyme that adds a phosphate group to something. Okay? So cyclin-dependent kinase and cyclin. All right. So you have two categories. You have two categories of genes that you need to know about. The first category, they are called tumor suppressor genes. Tumor suppressor. Tumor suppressor genes. And I don't think suppressor is spelled right. But is it two P's and two S's?
I think so.
Oh, okay. So tumor suppressor genes. These, the protein products, the products of these genes inhibit cyclin-dependent kinases. How do you make a tumor? Yeah.
You tell it to not stop growing.
Yes, the cells grow like crazy. Well, in order for the cells to grow like crazy, they have to have these cyclin-dependent kinases plus the corresponding kinase. Wow, the bells are still ringing early. It's 9:39, but the bell rang. So, all right, we'll have to finish this on Thursday. I guess our class did start a minute early, so it makes sense why it would end a minute early. Have a wonderful day. Enjoy the rest of your Tuesday.
You as 2 times teaching.
Yes.
So I have